Hey guys, so in this lecture we are going to be looking at plant genetics. And a lot of this is going to be an overview as we're going to see in the beginning. I've covered many of these topics already, so I'm going to kind of move through the first part of this lecture fairly quickly because we've gone over it, and then we'll get into some new stuff following that. Right, so if we're talking about plant genetics, naturally we are going to start with DNA, which is the hereditary material of all organisms, living organisms, right? And we've talked about DNA before. We know it carries the instructions for the development, functioning, and growth of organisms. It has this double helix backbone, this double helix shape, sugar phosphate backbone, and these nucleic acid bases. And it's the sequence of these nucleic acid bases that consists of adenine, thymine, guanine, and cytosine that ultimately determine an organism's genes. Right? And as we just tested on it, it was a question on your test. As we've learned before, a gene is a segment of DNA that codes for a protein. And your genes, in combination with your environment, ultimately are going to determine a trait, or the phenotypic trait, what we observe. Right? And as I mentioned before, it's the sequence of nucleic acid bases that we see here that is going to determine the particular protein that is made, that interacts with the environment, that produces a trait. And that process, which we refer to as gene expression, there's a couple steps to it, and it begins with that process of transcription, where our DNA is transcribed into messenger RNA, and then there's the process of translation, where this RNA, messenger RNA, transfer RNA binds to this messenger RNA. This takes place in that ribosome. And during that process, it creates this amino acid chain, which gets modified and folded onto itself to create a protein. Now, as we've learned before, we have organisms have what we call a genotype and a phenotype. And that genotype is the genetic makeup of the individual, the organism's blueprint, right? It consists of all those genes. The phenotype is the actual physical traits and characteristics that we observe, right? And that phenotype is based on the genes, the genotype, that and environmental influences. We talked about phenotypic plasticity in previous lectures, right? Okay, so this is a little bit new. I don't think I covered this, but your DNA is wrapped up and compressed into what are called chromosomes, right? And you have 46 chromosomes, right? The number of your chromosomes is the same in all of your vegetative or body cells, right? So most of the cells of an organism, except for the sex cells, they have the same number of chromosomes. And most organisms have 2 pairs of each chromosome. Right, and we refer to them as diploid, or 2N, that you see here. N standing for a single set of chromosomes, right? So you have a single set of 23 chromosomes and a copy of those, right, making of each one, which gives you a total of 46. Now, as I said, all of your body cells, and same if we're talking about vegetative cells of a plant, they will all have the same number of chromosomes. However, the sex cells are different. They are haploid. They only have 1 copy of each chromosome. That is because sex cells combine, right? You get your sperm and your egg, and they combine to make a new, eventually a new organism that is now complete, that has 2 copies, right? All right, so we have 2 copies. An organism has 2 copies of each chromosome in all of its cells. It's going to reproduce sexually. It produces sex cells. Those sex cells are haploid. They only have 1 copy. When those cells combine, they now are diploid again. Now, that is the case for a lot of our organisms, but sometimes, especially in our plants, we can have, due to mutations, which we'll talk about, we can have species that have extra copies, right? So they can be triploid, 3N, so they have 3 copies, tetraploid, pentaploid, hexaploid, etc. And this can result in different traits and different species that evolve. It's kind of interesting. But we refer to this as polyploidy. And I don't think I'll get into it in too much depth, but it's just something important to know because some of our agricultural crops are polyploids. So I mentioned that you have two sets of your chromosomes. So as a result, you have two copies of each gene, right? And that is true for all of our organisms that are diploid, right? So you will have pairs of chromosomes. So chromosomes that are the same are referred to as homologous chromosomes. And they will each have the same genes, but just different, potentially different variants of those genes, which we refer to as alleles. Right, so for example, if we're looking at a pea plant here, and on this pair of chromosomes, on the one chromosome, we have genes that record height and genes that record pea coat texture, right? And because we have two chromosomes, we have two different alleles or two different copies of those same genes. In this case, we have up here, both of these chromosomes have the same allele that is going to code for a tall pea plant. But they could have different alleles, and here one of these chromosomes has a gene that's going to code for a wrinkled seed coat, whereas the other has a gene that's going to code for a smooth seed coat. So what is going to be the phenotype, right? We have two competing versions of a gene. What is the actual phenotype? What is the actual trait going to look like? Generally, alleles are either dominant or recessive. And this isn't always the case. There are examples of codominance or incomplete dominance, but we're not going to get into that. What I want you guys to know is that generally you will have dominant alleles that are expressed whether the plant is homozygous or heterozygous, and I'll talk about that in a second. But essentially, dominant alleles, if an organism has a dominant allele, that allele is going to be expressed. And then you have recessive alleles, which are only expressed when both alleles are recessive. Right? So as an example, here is alleles that code for pea flower color. And this capital P is an allele that's going to code for purple flower color. This lowercase p is an allele that's going to code for white flowers. And here are different options for what the genotype could be, right? So you can have a genotype where you have two of the same alleles. Both are going to code for purple flowers. Obviously, you're going to get a purple flower. And we refer to when both alleles are the same, I'll show it in the next slide, we refer to that as homozygous. Now, you can have a situation here where one allele is for purple and one allele is for white, but because this purple allele is dominant, it is the one that's going to be expressed. So in a case where your alleles are different, and we refer to that as heterozygous, the dominant allele is going to be expressed, and that's going to be the phenotype. So you see that here, where these heterozygous genotypes. And you only get your white flowers, which is the recessive allele, when you have two recessive alleles. Right? So if they are homozygous recessive, right, both of our alleles are recessive, that's when you're going to get your white phenotype or your white flower. All right, and I already kind of mentioned it, but just to reiterate, when we are talking about alleles, and genotypes of alleles, right? We can have homozygous, where both alleles are the same, or heterozygous, where they are different. Now, when we are doing tracking genes or kind of doing breeding experiments, it is beneficial if we track the genotypes when we are crossing plants, right? Because if we track genotypes, it will give us an estimate of what we would expect the progeny to be in terms of their genotypes, and hence what their phenotype should be. Right, so the easiest way, and you've all probably have done this if you've taken a genetics class for sure, just through high school biology, you would have talked about genetics and you would have done Punnett squares. Punnett squares is just a nifty little tool that will help you determine genotypes of offspring of parents, right? If you know the parent's genotype. So, for example, we are going to cross two pea plants together. And we have our male up here and our female over here. And we know each of their genotypes. So each of them are heterozygous. Again, heterozygous means they have different alleles. So you have your one dominant allele here. In this case, this is going to be the capital B is now going to stand for purple. And lowercase b is going to stand for the white flower allele. So they're heterozygous. So they have each version of an allele. Their alleles are different. So when you're developing your Punnett Square, if you're just doing one trait like we're doing here, which is the easiest, you take your one parent and you put each of their alleles on the top, and your other parent and put each of the alleles on the side, and then you essentially just combine each alleles to determine what their offspring should be. Right, so you bring this B down and you bring this B over, and you would get a homozygous dominant offspring. You bring this B down and this B over, and you have heterozygous over here. You bring that allele down, that allele over into this box, and you have heterozygous. And then you bring that B down and that B across, and that gives you a homozygous recessive. So if you crossed two heterozygous plants for, in this case, flower color, your resulting genotype of your offspring should be a ratio of 1 to 2 to 1. Right? 1 BB, 2 heterozygous big B, little b, and 1 homozygous little b. Right? So that should be, through a big cross, if we're going to cross two of these plants and we get a whole bunch of offspring, those offspring should have genotypes in that ratio. And now if they follow normal, dominant, and recessive, your phenotype should be, what we actually see, should be a ratio of 3 to 1. Right? Because these heterozygotes are going to have the same phenotype as the homozygous dominant here. Right? So you should see 3 purple flowers for every 1 white flower. Right? So Punnett Square's kind of nifty way of being able to track the genotypes of offspring if you're doing crosses. This is more important when you get into breeding, and it gets way more complicated, but it's a good simple method to understand, because it's going to help you understand how genes and traits get passed on. Now, what I want to do is talk a little bit about natural selection, because natural selection is the process by which the diversity of life was created, right? So all the organisms that you see, really up until the time of humans started breeding experiments and doing artificial selection, which I'll talk about, but natural selection essentially shaped all the organisms around, including our crop species, or at least the ancestors of our crop species. And it's a hugely important process, and by understanding natural selection, it gives us a better understanding of how, when we have done artificial selection or breeding experiments, how those have worked, because we essentially just hijack this system. So by understanding the components of natural selection, it should help us better understand artificial selection. So that's why I want to talk about it. Also, I want to talk about it because it's just really cool. It's super important. So, for natural selection to occur, it requires 4 things. First is you need variation in a trait. So, for example, we're going to stick with flower color, and we'll stick with pea flower color. We have variation if there are different types of that trait. So, we have purple flowers and we have white flowers. That's variation. Second, you need heredity of that trait. So, that trait needs to be able to be passed down to offspring, which occurs, as we've seen, through those genes that code for different flower color, right? That's heredity of a trait. So you need variation of a trait, and you need heredity of a trait. Then you need variation in reproductive success. And what this means is that we have offspring that have different traits. Some have purple flowers, some have white flowers. The plants that have one trait or the other need to be more successful in producing offspring than the other plants. Right? So hypothetically, we will say that the purple flower plants are more successful at producing more plants. Right? That would be variation in reproductive success. And then lastly, you need a non-zero correlation between trait and reproductive success. This is essentially just a fancy way of saying some of those traits produce more offspring than others, right? So variation in reproductive success just means that some organisms produce more offspring than the others, and then that non-zero correlation means that it's related to that trait. Really, that's really what that's saying. So, like I said earlier, the purple flowers produce more offspring than the white flowers. So if you have all four of those things, you'll have natural selection in action, and through time, you will have a species that modifies, right? Or population, I should say, that modifies. And that population may turn into an entirely new species, given enough time. Okay, so the first is variation in a trait. We need to have members of the same species and members of the same population that have variation in whatever trait it is, whether it is leaf shape or flower color, right? So where does this variation come from? And ultimately, where this variation comes from are mutations, right? It's coming from mutations. So there are a number of different types of mutations that can occur. But essentially what you need to know is when your organism is going to reproduce sexually, when it's going to create sex cells, it goes through this process of, initially first, of DNA replication. So each of these chromosomes creates another copy, though that cell then splits, right? So these each create another copy like that. That cell then splits. We refer to this process as meiosis 1, and then it splits again to create sex cells. Well, during this process of DNA replication, sometimes there are mistakes. That's essentially what mutations are. They are changes in DNA sequence that occur during this DNA replication. They're essentially mistakes that happen, right? And these mistakes, there are a number of different types that can occur, right? So sometimes, if this is the normal sequence, right, of our nucleic acid bases, C, T, A, G, if we're trying, during this process of replication, we're trying to make another copy of this, sometimes a random nucleic acid base can be inserted, right? That's an insertion mutation, right? So here a G was inserted, and now we have a new sequence. Sometimes it can be removed. Sometimes there can be a duplication, so a whole section can be duplicated. In the case of CT here, that gets duplicated, and now you have that occur first. Or there can be an inversion where the order of these bases switch. Right? And this can occur with a sequence of bases. It can occur with whole chunks, whole sections of DNA. All of this will create, all of these are mutations that potentially can create variation in a trait. Now, a lot of these mutations are negative, right? They are not positive. They won't lead to, well, they can create variation, but it's not good. Many mutations have no effect. And then there is a small percentage that are positive, that lead to what we'll see later, increases in fitness, or you're able to produce more offspring. Oh, and here we go. And besides looking at these different types of mutations here, we can classify mutations in terms of, like I said, a lot of them are neutral, they have no effect. Some are harmful, and some are beneficial. And when I mentioned fitness before, that is, excuse me, the organism's reproductive success, right? So its ability to produce offspring and how many offspring it can produce. Right? And as I said, most mutations are neutral or harmful. So in the beginning, I talked about polyploidy, right? And that is an instance where there's an error during that meiosis. So if we go back, that process of meiosis, you have this initial DNA replication, and then those copies get broken down into two separate cells, and then they get ultimately broken into four separate cells, so each one has just a single copy. If polyploidy that doesn't occur, right? So for whatever reason, sorry, I'm coughing. For whatever reason, these chromosomes don't separate. So you get an additional copy of chromosomes when you combine them into an egg cell. So instead of having your normal diploid cell, you essentially have two diploids now that are going to fertilize, and now you have a tetraploid. So it has 4 copies of each chromosome. And this results in different traits that we could see. And there are examples of polyploid plants in our crop species, some very common ones, right? Wheat, tobacco, potatoes, bananas, boysenberries, strawberries, all have had these polyploidy errors. So they all have more copies of chromosomes than standard organisms that have 2 copies, right? And at an extreme end are ferns, which can have up to 400 copies of chromosomes, which is insane. But just something interesting, and this process can result in very different traits than the normal species. So, variation in trait, right? That's the big one. Heredity of trait, we've already talked about. That is how our traits are passed down in genes, right? We've mentioned about that. So, like I said in the beginning, for the vast majority of history, a natural selection has shaped organisms, up really until humans, where we have co-opted this process. And where we influence this, we refer to it as artificial selection. And for most of human history, this artificial selection has been done through breeding. So this is a process where we select a female plant and a male plant, or if we're talking about organisms, a female and a male animal, that have desirable traits, and we cross them together to produce offspring that have those desirable traits or increased traits, right? And in plant propagation, if you guys decide you want to take that, we go a little more in depth into breeding programs, and I give you some kind of cool examples. But if we, again, now are looking at our natural selection, what is breeding doing? Well, we are greatly influencing reproductive success, right? And this correlation. So we are taking the organisms that have traits that we like, and we are crossing them, right? So they are going to have huge reproductive success. Any organism that has a trait we don't like, we don't cross it. So by influencing these 2 processes here, we can greatly speed up the process of evolution, and that's really what we're doing here with artificial selection. And it results in all these different varieties and variations in varieties that we have today. Right? For example, here is wild mustard. This is a single species, wild mustard. All of these species that you see here are all the same, right? And they've all derived from mustard. We have just bred them for different characteristics, right? So with cauliflower, we bred wild mustard to have sterility of flowers, and it results in cauliflower. Broccoli, we bred for suppression of flower development, which gives us the characteristics that we see in broccoli. Cabbage, we bred for suppression of internode length. So again, those nodes are those spaces, or internodes. Nodes are where you have a stem or a flower that would branch out, or a bud, and internodes are the space between them. We suppress that, and that results in cabbage. Kale, an enlargement of these leaves. Kohlrabi, an enhancement of the lateral meristems, right? So all of these are the same species, and they're all essentially related to wild mustard, and we have just, through generations, bred these plants to produce traits that we wanted, and essentially vegetables, different vegetables that we see here. All right, so that video, I'll put the link in, I guess, in the description. That takes you to another video that shows wild ancestors of some of our common crop plants. It's pretty interesting to see what the original version of fruits and vegetables looked like before we modified them. So another important thing that I want to talk about in regards to artificial selection is this process called hybrid vigor or heterosis. I want to talk on this because it is so commonly used in our corn. So hybrid vigor is this unique result that occurs in which hybrid offspring or hybrids have improved function over their parents, right? And essentially our hybrids here, when we talk about them, they are heterozygotes, right? Talking all the way back from the beginning of this lecture when we looked at a single genotype of a single gene, right? And we have those two alleles, and a heterozygote is one in which we have different alleles. Right? And that's really what's happening with our hybrids that we see here. You take one parent that is homozygous for traits, you take another parent that is homozygous for traits, and you cross them together to create a bunch of heterozygotes. And those heterozygotes, or those hybrids, have improved function over both parents. And we see that with corn. Almost all corn that is planted is hybrid corn. And it's because these hybrids have so much better function than parents. Look at the size of them. Their growth, their yield is greater than either parent. Right? If we look at corn cobs, look at the size, the amount of kernels, etc. All is improved, yield is improved with hybrid corn. Now, because these are heterozygotes, you can't collect the seeds from their cross, right? When these produce corn kernels, you can't collect these seeds and then plant them, right? It's due to the genetics. Let's see if I got oh, perfect. Oh, good, I go into it right here. You can tell I previewed this PowerPoint before talking. So if you look at a cross, right, as I said, our hybrid corn are crosses of two inbred lines of corn. So they are homozygous for traits, right? So they are, for, say, a given trait here, hypothetical trait, one line, one parent will be big A, big A. The other parent is little a, little a. Right, so if we cross those, right, we're going to create a bunch of hybrids that we see here that are heterozygous. Right, and this is all the corn that we plant grows big. We get a lot of yield, a lot of corn cobs. Now, if we save some of that corn and we go to plant it, we are now, these were all fertilized. These were all hybrids that fertilized themselves, right? So the resulting offspring are not all going to be heterozygous, right? If we cross two heterozygotes together, we get this genotype combination of big A, little a, big A, little a, big A, little a, little a, right? So only 50% of a cross of our two heterozygotes are going to be heterozygotes as well. The other two are going to have the same genotypes as the parents, and those are the lesser yielding corn, right? So it's why instead of saving your seeds, your corn seeds, and planting again, most farmers will just go off and buy new hybrid corn seeds, because if you save your seeds and get a cross, you're only going to get 50% of your next generation that turns out being hybrid or having the same genetics as that hybrid corn. And if you save that and cross that later, you can get into some difficulties as well, right? So by buying new seed each time, you're ensuring all of your corn are going to be hybrid and they're all going to have those traits that you want. Now, we have advanced enough in our science and technology that we have more advanced ways to genetically modify organisms. that process of artificial selection, that is a process of modifying organisms genetically, right? We are shaping their genes. We are just doing it through breeding, which is what you can maybe want to consider a more natural process, right? We're speeding up the process of evolution. We're kind of altering it, but we're doing it through what are otherwise normal processes that occur. With genetically, when we talk about nowadays with genetically modified organisms, what we are talking about is an organism whose genetic material has been modified using some sort of genetic engineering techniques, right? And there are a number of techniques that they use, and they're becoming much, much more advanced than initially, right? So initially, one of the first techniques is that we would just bombard our plants with radiation, right? We would shoot gamma rays at them, X-rays at them, and those rays will induce mutations, right? It's why X-ray techs have that big lead shield, right? It's to prevent those X-rays from impacting their cells and producing harmful mutations over time. But we just did this with our plants, right? We would bombard them with radiation, and sometimes there would be a beneficial mutation that would occur. Remember, like most mutations that occur are either neutral or harmful, but sometimes you get a beneficial one. Right, and as examples, here are some crop varieties or mutations that have occurred that we were able to create through this process. Right, so with different rapes or rapeseed, flour, sunflowers, by exposing them to radiation, we got different traits, whether it is skin color in apples here, or cooking oil quality in some flax, a dwarf high-yield variety of rice, etc. Spineless pineapples. Right? So this is kind of the first thing that we did to genetically modify organisms. We just shot them full of radiation, and sometimes they produced beneficial mutations. And as we have advanced in technology and our understanding of science and technology, we have started developing more elegant methods for modifying organisms, right? We have transgenic crops now, where you implant the gene or genes from different organisms into a plant. Recently, the newest development is CRISPR-Cas9, which is allows you to be more targeted than this process up here. And that really allows you to really insert or remove specific DNA sequences. So it's a very, very targeted process, more so than traditional methods. Anyway, if you are interested, I'll include these links in the description. They go over each of these processes and give you a little overview. It's interesting stuff for you guys to know. I'm not going to go over it in this lecture or make you know it, but it is interesting, and I would suggest you watch the videos. And that is it. So, like always, any questions on this stuff, let me know. I hope you guys are doing well, and I will see you in the next video.